Hello, everyone. Welcome to the Ethel Browne Harvey Postdoctoral Seminar Series. My name is Yinan, and I'm a postdoc at the University of Basel and will be moderating today with Keng Vong, a postdoc at UCSD. We're excited to highlight the work of our outstanding postdoctoral members. Today, Joshua York from the Northwestern University and Diana Fulmer from University of Pennsylvania will share their research. Each speaker will give a 20-minute talk followed by 10 minutes Q&A. Please enter your question in the Zoom Q&A box. I will introduce the first speaker. Joshua York completed his bachelor and master degrees from the University of Central Oklahoma and did his PhD in biology at the University of Oklahoma. From September 2020, he has been a postdoc scholar in Carola Vong's lab at Northwestern University studying the development and evolution of neural crest cells. He has received the prestigious LSRF postdoctoral fellowship for his postdoc work. Today, he will talk about shared features of blastula and neural crest stem cells evolved at the base of vertebrates. Joshua, the stage is yours.
Thank you, Yinan, for the intro. Share my screen here. Okay. Again, thank you for the introduction. Thank you to SDB for giving me the gracious opportunity to present my work today. Today, I'm going to be talking about some of my work looking at the evolutionary origins of the neural crest and neural crest developmental potential. So vertebrates are a highly diverse, ecologically and evolutionarily successful group of animals. And ever since making their first appearance in the seas of the Cambrian area nearly 500 million years ago, they've had a remarkable history, branching out into tens of thousands of different species that have colonized every continent on the planet, ranging in size from a few millimeters to over 30 meters in length. It's this great variety in vertebrate form and function that's really prompted one of the big questions in evolutionary biology, and that is simply, where did vertebrates come from? What are our evolutionary origins? It turns out the answer to this question lies in understanding how vertebrates undergo the process of development from a single cell to a multicellular organism. And the reason for this is that a lot of what distinguishes vertebrates like ourselves from our closest invertebrate relatives is a small group of cells known as the neural crest. So neural crest cells are a migratory embryonic stem cell population that give rise to a multitude of diverse cell types and structures that really define much of what it means to be a vertebrate, right? Things like the cartilage, bone, and muscle of our head, face, and neck, colorful patterns of pigmentation in skin, feathers, and scales, most of our peripheral sensory nervous system that allows us to interact with our external environment, and even things like parts of our heart and teeth. And so what this really highlights is my overarching question, which is, how did this broad developmental potential evolve in our earliest vertebrate ancestors over 500 million years ago? So this question is interesting, it's important, it's also a little controversial over the years because for a while, neural crest cells seem to violate one of these mainstays of developmental biology theory, this idea of progressive lineage restriction over time. So what I mean by that is that neural crest cells appeared to be more stem cell-like than the ectodermal progenitors from which they're apparently derived, in effect rolling back up Conrad Waddington's famous developmental landscape here, before descending once more to give rise to specific cell types and structures. And so to try to reconcile theory with evidence, we in the Laban lab have put forward this alternative model called the retention of pluripotency model, which states that neural crest cells aren't induced to become pluripotent, but they are pluripotent by virtue of maintaining part of the gene regulatory network underpinning blastula stage pluripotency, that earlier phase in development. And by doing so, this endows these cells with the ability to give rise to cell types associated with mesoderm, ectoderm, and even in some cases, endoderm. This has only really been described thus far in traditional jawed vertebrate model systems, including the workhorse of the Laban lab, the African clawed frogs, and Epislavis, a member of the jawed vertebrate side of the tree, making it unclear whether or not this is an evolutionarily conserved feature of neural crest biology across vertebrates. So to try to get at that question, we have to look to the other major lineage of vertebrates, represented here by the jawless cyclostomes, represented here by this face only a mother could love, the sea lamprey, Petromyes and Marinus. So what are lampreys and why are they important? Well, lampreys are a group of primitively jawless vertebrates that, along with another group called the hagfish, they together constitute literally the only surviving relics of an ancient but ecologically dominant group of jawless fishes that were among the first vertebrate animals to evolve on this planet well over 500 million years ago. And that makes them a really attractive model for these types of studies, right? Because what we can do is we can study the developmental mechanisms controlling formation of neural crest cells, blastula stem cells, and so on, the GRNs underpinning their formation, and then compare and contrast that to the jawed vertebrate side of the tree with a frog like Xenopus. And what this allows us to do, this classic comparative method in evolutionary biology, is basically work backwards in time to stitch together a picture of how these cells ultimately evolved their impressive developmental potential. And so using this strategy, my question today here is pretty straightforward. Do we see evidence that lamprey neural crest cells also form by retaining the signature expression and functional roles from much earlier in development, the blastula? And so if you have this question in mind, you can make a couple of predictions. The first of which is that what I'm calling the neural crest GRN, right, should be expressed much earlier and active much earlier in development in animal pulse cells of the blastula. So the question is, do we see evidence of that in both species? And indeed we do. So if we look at classic neural crest regulatory factors like AP2 alpha, ZIC1, snail, FOXD3, and many others, I'm only showing you a few of these today for the sake of brevity. In Xenopus, these are expressed in animal pole blastula cells alongside core members of the vertebrate pluripotency GRN, factors like c-Myc, KLF, SOXB, and many others. And crucially, we see this in lamprey as well. Not only are these genes encoded in their genome, but they show a striking pattern of expression that's similar to that of Xenopus. We see AP2 alpha, ZIC-A, homologs of these genes in Xenopus expressed in animal pulse cells, again, alongside core members of the vertebrate pluripotency GRN. We can use more modern techniques like HCR FISH. So here you're looking at the animal pole view of a lamprey blastula, and we can show nice cellular level colocalization of homologs of SOX2/3 in lamprey called SOXB1A, and these colocalize with PAX3 protein, a bona fide marker of the neural plate border neural crest invertebrates. So the second prediction we would make is that these genes shouldn't merely just be expressed in the blastula or co-expressed, but they should resolve, right, spatially and temporally into the neural crest forming regions. And indeed we see this. So if we take a canonical neural crest regulatory factor like ID3, which we know is essential for neural crest formation, in Xenopus and lamprey, with the homolog of this being ID-A, they're both expressed in the blastula. They're both retained into the gastroectoderm, resolving nicely to these lateral strips called the neural plate border, the neural crest progenitor domain, and then ultimately in these paired strips of the neural crest in Xenopus, and in lamprey, which neurulates into the dorsal neural tube, ID expression there as well. And crucially, we also see this for pluripotency associated factors. So KLF17 of the recent, of the KLF2417 clade transcription factors, again, is present in both Xenopus and lamprey, and they show this striking pattern of resolution ultimately into the neural plate border and the neural crest of both species. And again, we can use more modern techniques, again, like HCR FISH. We're looking here at a neural plate border stage in a lamprey embryo. We see PAX3 expression in the neural plate border, SOXB1A expression in the neural plate extending laterally into the neural plate borders, and then we can see cellular level colocalization for these factors. And then again, after neurulation in the dorsal neural tube, we have pre-migratory neural crest cells here co-expressing PAX3 and a pluripotency associated factor, KLF17. So what this suggests is that this signature from the blastula, where you have co-expression of neural crest and pluripotency factors in the blastula and animal pulse cells, they resolve spatially and temporally, ultimately into developing neural crest cells. This is conserved across jawed and jawless vertebrates, suggesting that this is an ancient mechanism controlling the formation of neural crest stem cells. So now I want to change gears a little bit and move from this candidate gene approach into a genome-wide approach using comparative transcriptomics. And so what we did here is we manually dissected animal caps from lamprey blastula embryos. For anyone that's worked with lamprey, you can hopefully appreciate how difficult this is. It took a lot of time to figure out, but we managed to do it. So we cut animal caps from them. We did the same thing for Xenopus. And then for the neural crest stages, we integrated our lamprey blastula data set with data sets from the Saka Spengler laboratory, in which they manually dissected neural crest cells. For frogs, we did something a little bit different. Frogs have this amazing technique where we can inject Wnt8 and chordin mRNA at the 2-cell stage. We culture those to blastula stages, cut the caps, and then they take on temporal progression and forming neural crest cells identical to that we see in the whole embryo. So we can get this nice homogeneous population of cells for these animals this way. And so what I want you to pay attention to is not necessarily the individual gene names, which are difficult to see, but rather the color coding. So we did two important things here. One, we Z-scored within species. This gets around the thorny issue of trying to reconcile which genes are which between species separated by vast evolutionary distances. The second thing is we compared within and between cell types across species. So I want you to focus on the color coding, particularly for the blastula stages, which show this remarkable degree of conservation of the relative transcriptional abundance of each of these components with respect to one another. We do see conservation somewhat in the early and late neural crests, but less so compared to what we see in the blastula, where it's very strong. So much so that despite being separated by 500 million years of independent evolution, we can still detect positive cross-species correlation coefficients in terms of the relative transcriptional abundance of these components. So for example, we can take the TPMs for Xenopus AP2 and compare that to lamprey AP2, SOX-E factors to SOX-E factors, and so on. And what you're finding is that strong natural selection pressures have likely maintained these factors at similar levels across deep evolutionary time. And when you look at the genes that fall in the best fit line here, they're genes that make sense that they would be the strongly conserved genes within these GRNs, genes like KLF, AP2, ZIC, SOX-B, and many, many others. So now I want to change gears and talk about, or excuse me, we can also detect this in terms of the temporal trajectory of these genes. So we can not just look at individual regulatory factors, we can look at the temporal dynamics of clusters of these genes and how they change over time. So we can see, for example, GBX2, PRDM1, Wnt8, in lamprey and Xenopus, and these clusters show conserved trajectories. We can see the same thing for SOX-E factors, AP2 beta, CYP1, ZEB2, and many, many others. So it's not just one-to-one basis, it's that clusters of putatively co-regulated genes show these similar temporal dynamics over time. Okay, so what I've shown you up until now is largely evolutionary conservation, right? Similarities in terms of how are the GRNs similar between the blastula and neural crest, and how are they similar across species? Now I want to talk about the differences. So after all, it's reasonable to expect that the neural crest GRN evolved to become distinct in many ways from this ancestral pluripotency GRN, in part by co-opting novel genes and signaling pathways downstream of that GRN. So what are those novelties and how can we find them? So the answer is through differential expression analysis. So what we can do here is identify genes that are not expressed in the blastula and then significantly enriched in the neural crest. And when we do that and compare and contrast between species, we find that this GRN can be broken down into these two temporally distinct modules. So the first module are genes from the blastula, so genes like ID, MYC, SOX-B, and others that are expressed early in development and then retained into developing neural crest cells, as I showed earlier. And then this analysis uncovers a second module, which consists of a cohort of genes that are specific to neural crest cells. And these genes occupy three really interesting categories. So genes like GBX2, IRX, and Wnt signaling are responsible for spatially patterning the vertebrate and ectoderm into these distinct territories. Genes like PDGFRA, AP2 beta, CYP-ZEB, these genes are responsible for epithelium to mesenchyme transition in long distance migration. And then genes like SOX-E factors and the vertebrate-specific endothelin signaling pathway control lineage diversification of these stem cell states into derivatives associated with mesoderm, ectoderm, and endoderm. So we think that the neural crest GRN evolved by elaborating upon this ancestral GRN by co-opting novel genes downstream of that GRN. So now I want to talk about what I like to call an interesting evolutionary case study involving the OCT4, now that we know called the PAL5 family of transcription factors, in the role and formation of neural crest in pluripotent blastula stem cells. As many of you know, OCT4 is a founding Yamanaka factor. It's essential for reprogramming somatic cells back to this pluripotent state, but it is just one member of this large pluripotency GRN. So why focus on this one? Well, for a very good reason. For about the past 20 years, it's been thought that this gene was simply absent from jawless vertebrate genomes, and recent work from the Osaka laboratory showed the importance of these neural crest cells because they give rise to most, if not all, of the neural crest-derived craniofacial structures in jawed vertebrates. So you can infer, of course, the evolutionary significance of this observation. If jawless vertebrates lack OCT4, and OCT4 is known to be important in formation of the craniofacial like the mesenchyme and novel morphological structures that jawless vertebrates lack, this could be an important evolutionary step. However, our RNA-seq analysis in lamprey uncovered this really interesting uncharacterized transcript, as it was called at the time, that was among the most top 10 highly expressed genes in the blastula, and then just nothing into the neural crest. As you can see here, TPMs are hovering around 0 in the neural plate border and the neural crest. The top hits from a blast analysis of this transcript were OCT4 PAL5 factors in jawed vertebrates. This was one of the most exciting moments of the project, uncovering this new gene, new gene. And we confirmed this with phylogenetic analysis and chromosomal synteny comparisons, indicating this is a bona fide ortholog of PAL5 in jawless vertebrates like lampreys, and it's also been uncovered in hagfish. So phylogenetically, it's not just PAL5, it acts like PAL5. So if we do co-immunoprecipitation experiments, looking at the ability of these proteins to bind together, we can see that lamprey PAL5 is capable of physically interacting with lamprey KL17 and lamprey c-MYC over here, similar to what we see for PAL5 in jawed vertebrates. Naturally, we wanted to know where it was expressed, and confirming the RNA-seq data, we can see robust expression of lamprey PAL5 in animal pulse cells at the blastula, and then we don't really see any expression after that into the developing neural crest. So if you compare the situation to Xenopus over here, we see that there are 3 copies of this gene, PAL5 F3.1, 2, and 3, issued by tandem gene duplication tens of millions of years ago. There's evidence for subfunctionalization of expression going on, but overall, the combinatorial expression profile is consistent with retention from the blastula into the neural crest. And we can, again, use modern techniques like HCR FISH to show nice co-localization at the cellular level for neural crest markers like SNAIL2 with PAL5 transcripts. So lampreys, like other vertebrates, have PAL5. Like other vertebrates, it's expressed in blastula cells. Unlike other vertebrates, they don't retain it or reactivate it in the developing neural crest. So that presents us with two interesting evolutionary hypotheses. So one, lampreys never had it, jawless vertebrates never had it in the neural crest. So that means PAL5 co-option with the neural crest is an innovation of jawed vertebrates. Alternatively, PAL5 expression was present in the neural crest of ancestral vertebrates, but then lost secondarily in the late Anglia to modern lampreys. So in thinking about these hypotheses, we reason on the basis of number two, if it was lost, then maybe the lamprey PAL5, even though it's not active endogenously in the neural crest of these animals, still retains some latent capacity to engage the neural crest GRN. Well, that's a perfect experiment for our lab because we can stick lamprey PAL5 in a frog and see if it's capable of expanding neural crest cells. And that's exactly what it does. So if we stick lamprey PAL5 in there, we get expansion of PAX3, ZIC1, and SNAIL2 in a manner that's virtually indistinguishable from that of doing the same experiment with Xenopus PAL5 expressed at equivalent levels matching that of lamprey PAL5. So PAL5 is capable of promoting neural crest formation in Xenopus. We complemented this with a loss of function approach. So is PAL5 required for neural crest formation? And it is, because if we use morpholino-mediated gene knockdown targeting the two zygotic PAL5 paralogs, we see a near total loss of neural crest gene expression on the injected side. And providing further evidence that this is a functional PAL5, the lamprey version of this gene is capable of fully rescuing these neural crest depleted phenotypes in a manner that's virtually indistinguishable, again, between the Xenopus rescues. So next we wanted to ask, how did PAL5 evolve this critical neural crest enhancing activity in vertebrates? To try to answer this, we revisited our phylogenetic tree, which suggested that PAL5 shares a common ancestor with PAL3. And this is consistent with previous work showing that PAL5 likely evolved from a PAL3-like ancestor, which raises the question, what do PAL3 genes do? They're largely responsible for promoting neurogenesis in the central nervous system and peripheral nervous system. The question overarching here, though, is PAL3 also capable of promoting neural crest formation, or is this a synapomorphy of the PAL5 clade that is specific to vertebrates? And it appears to be specific to PAL5, because if we overexpress Xenopus PAL3 or lamprey PAL3, we don't promote neural crest formation, we actually inhibit it. And consistent with this early role in promoting CNS and PNS neurogenesis, we see an expansion of SOX3 from the neural plate into the developing neural crest region. But this is not a general ectodermal expansion because we're actually losing epidermal progenitors. So PAL3 seems to have this ancestral function in neurogenesis and does not have the ability to promote neural crest formation. So finally, we asked if PAL5 ultimately evolved from this PAL3-like ancestor, does it retain this function to promote neurogenesis in addition to neural crest enhancing functions? And again, indeed it does. So whether we're looking at lamprey PAL5 or any of the three Xenopus PAL5 paralogs, they're all capable of promoting expansion of neural stem cells in addition to neural crest stem cells simultaneously. And again, this isn't just a general ectodermal phenotype. It's specific to neural crest and neural stem cells as we're losing expression of epidermal keratin. So the final thing I want to talk about is, why does PAL5 do this but not PAL3? What explains some of the differences in their differential ability to promote neural crest formation? So towards the end of the paper, we delved into a little bit of a sequence analysis comparisons between these families and found this really interesting extended C-terminus region of PAL5 that's conserved largely across jawed vertebrates and to some extent jawless vertebrates. It's not present in PAL3. And then likewise in the conserved homeodomain and PAL-S domain, there are residues for simulation and MAPK phosphorylation sites that are present in PAL3, but not PAL5. So one of the hypotheses that we're working with now is that protein sequence evolution drove functional evolution, explaining differences in the divergence activities of these proteins in the neural crest region. And so current experiments that we're working on are creating protein chimeras where we swap these domains, we delete domains and so on to try to pin down which of these residues and which of these domains are critical for PAL5 function. So finally, I put this all together into a model to explain the origin and evolution of neural crest developmental potential. So in the last common ancestral node point here of invertebrate chordates and vertebrates, we can be confident in at least two things. So one, these animals didn't have neural crest cells because it's a vertebrate innovation. And two, really, they probably didn't even have a functioning pluripotency GRN. So why would I say that? Well, there's two lines of evidence. One, Ventex, Nanog, and PAL5 have not been uncovered in invertebrate chordate genomes. These are vertebrate novelties. And number two, in our closest invertebrate chordate relatives, the tunicates, those animals have cells that are largely specified to a single fate shortly after the blastral stage and prior to gastrulation, suggesting that perhaps somatic pluripotency may be an innovation of vertebrates as well. So two big changes when we get to the last common ancestor of all vertebrates here. Number one, we see the evolution of neural crest cells. We also see the evolution of the full complement of pluripotency factors, the Yamanaka factors. And we think that those two GRNs co-evolved together originally in blastula cells and were retained in one way or another into developing neural crest cells, endowing these cells with their broad developmental potential. And then we have another significant event in the split between jawless vertebrates and jawed vertebrates. We think that PAL5 may have been lost from the neural crest of these animals and retained or reactivated in one way or another in the neural crest of jawed vertebrates. And this is evolutionarily significant, we think, because the absence of PAL5 from jawless vertebrate neural crest cells may explain the relatively restricted developmental potential of these cell types compared to that of their jawed vertebrate counterparts. And so with that, I would like to say thanks to my funding sources, LSRF and the Walder Foundation, Northwestern, as well as, of course, SDB for having me on here to talk about my research. I really appreciate it. I really appreciate everyone for coming out today and hearing about my work. The paper was published this past fall, so if you want to hear more about it, please take a look online. And with that, I would like to say thank you for listening, and I'm happy to take any questions.
Thank you, Joshua, for the wonderful talk. Yeah, the audience, you can start typing your question into the Q&A box of the chat. Maybe I start with one question. Is it only PAL5, or you also found other genes that are like exist in the blastula but not in your crest? Is it a network of genes?
For lamprey?
Yeah, for lamprey.
Yeah, so there are quite a few, and so we're trying. PAL5 was sort of the low-hanging fruit. It's an important gene. People know a lot about it. It's obviously important for pluripotency. But yeah, there are several differences, and that's kind of the focus of future work, is to figure out, okay, here are all the similarities, here's what's conserved, but differences, I think, in evolution are what are really important, because that's where you start to see differences in GRNs that correlate with differences in morphological structures, cell types, and so on. So yeah, we're trying to delve into that currently for future work.
Crystal Rogers has a question. This is an awesome project. Do you have evidence that specific neural crest derivative lineages are lost if you inhibit the PAL5 in jawed vertebrates, and if you add it to lamprey? Can you expand cranial?
Yeah, so that's a very good question. So yeah, so the paper I was talking about, where I had the quote talking about OCT4 positive cells give rise to most, if not all, craniofacial structures. So a lot of the craniofacial ectomesenchyme, especially the bony stuff, right? Those are not present in the craniofacial skeleton lampreys, which are only cartilaginous. So the experiment that we're planning on doing this summer is to do PAL5 gain of function in lamprey, and to try to get at that idea of if we can see changes in the GRN of these cell types by overexpressing it where it normally isn't. So yeah, no, that's definitely a cool experiment that we're doing this summer when we get lampreys.
Cool. Thank you. Carrie Adler has a question. Do you know if the targets of PAL5 are similar in lamprey and vertebrate?
Another excellent question, yeah. So this was sort of the first sort of skimming of these topics in lamprey. It hadn't really been addressed. Most people hadn't even really done blastula stage in situ. So I mean, that's sort of where it was at the beginning of this project. So the short answer is we don't know. But ChIP-seq is something that we want to try, for example, to look at binding of PAL5 in lamprey. But not just binding of PAL5 in lamprey, but maybe heterologous experiments where we take the lamprey PAL5 and do ChIP-seq, for example, in Xenopus, and see if it's capable of binding the same sites and which sites they bind that are different between species as well. So yeah, ChIP-seq is something we want to do. It's a little difficult with lampreys because, if there's any lamprey people here, you can appreciate the fact that we have basically a month and a half out of the year to get all of our experiments done. So we would have to prep a while ahead to get ChIP-seq experiments done, but that's definitely something in the future that we are wanting to do, yeah.
Thank you. Robert Cornell has a question. Hi, Josh, does your model predict that Yamanaka transcription factors bind the same site in blastomeres and neurocrest?
Yeah, so I mean, that would be an interesting observation to get at your question, about the differences. So I think what we'd expect is a lot of overlap in the binding of regulatory targets. And there's a former postdoc in the lab, Betsy Schock, who's now started her lab at the University of Kentucky. And she did this with SOX-B factors, showing that you have binding of SOXB factors to very similar sites for pluripotency factors and neural crest factors. In some cases, these are repressive. In some cases, these are involving activation. So I would predict a lot of overlap in terms of what they're binding to and how they're regulating them, although that could be differences in terms of negative regulation or positive regulation between those cell types. But I think in our model, yeah, I would predict that there should be significant overlap in terms of target cohort of genes being bound by PAL5 in both cell types.
There will be overlap.
Yeah.
Billy Swala has a question. Great talk, Joshua. I'm interested in the comparison of proteins with the lancelets and tunicates. Your work suggests that genome duplication that occurred in vertebrates may have allowed the evolution of neural crest.
Yeah, I mean, this is a classic question in the field of neural crest biology. It depends, I guess, on what your interpretation would be of tunicates, for example, which are argued to have neural crest-like cells, right? So, if that's the case, if they have a lot of similarities to neural crest cells, they function like neural crest cells, they lack sort of this very broad ability to contribute to diverse cell types, that would argue that basically most of what we call a neural crest cell would be independent of genome duplications. But I think a lot of this sort of sophisticated GRN architecture that we see in neural crest stem cells, particularly the multipotency, may have been driven by genome duplications. But yeah, it's definitely a contested question in the field. Yeah.
Ryan Hall has a question. Have you looked at the trunk neural crest and TNC cell types, and could cranial and trunk crest have evolved separately?
Yeah, it's a good question. So with lamprey, it's very difficult to do. This would be more amenable, I think, to something like a single cell experiment. So the cranial region of lampreys, pre-migratory neural crest cells, are pretty easy to dissect, or relatively easy to dissect. The trunk region is very narrow, very small, particularly at stages that are relevant to us. The Bronner laboratory and others have shown that the AP restriction of these domains is less complex than that of higher jawed vertebrates. So they putatively lack a vagal neural crest, for example. In terms of the AP differences, yeah, there's a lot of open questions about what GRNs are different between cranial versus trunk, and so on, but I haven't looked into that. From the pluripotency factor perspective and neural crest genes that we've looked at, a lot of those seem to be not really axially restricted. They're present along the anterior posterior axis.
Thank you. We have some fun questions here. Marsha and Laura asked, "Who's your lab artist?"
Yeah, that's my son Elijah. So for those of you who can't tell, this is of course a frog. This is a lamprey. This is a fish that they're parasitizing on. This is blood. And here's the E, which has the teeth in the sort of circummoral region of the mouth. So yeah, this is my son Elijah. He drew this for me.
That's really cute.
Thank you.
Let's see if there are more questions coming. If not, I think we'll stop here and pass it on to the next speaker. Thank you, Joshua, for the wonderful talk again.
Thank you, and thank you for the great questions. I appreciate it.
So today we are very happy to have Dr. Diana Fulmer from UPenn here today to talk about her work on cardiac neural crest cells. Dr. Fulmer received her PhD training at the Medical University of South Carolina in the lab of Dr. Russell Norris, and continued her postdoctoral training at Penn in the lab of Jonathan Epstein. Dr. Fulmer has received several fellowship awards, including the very prestigious Career Transition Award from Additional Ventures. So we are very excited to have you here today, and let us welcome Dr. Diana Fulmer.
Okay, get me shared here. Thank you so much for that wonderful introduction. And thank you to SDB for giving me this opportunity to come and speak to you today about some of my work that I'm doing in Jonathan Epstein's lab. So the Epstein lab is really interested in nuclear organization. And I know when I say nuclear organization or talk about chromatin inside of the nucleus, a lot of you may have an idea that looks something like this, where chromatin is just kind of haphazardly thrown into a big pile on the inside the nucleus, but we know that's actually not how chromatin is arranged. It's actually arranged into functional compartments, and we can see a great example of that in this electron micrograph. So you'll note in this image here that there is a lot of light gray, fluffy euchromatin localized towards the interior of the nucleus. However, the darker dense, transcriptionally silent heterochromatin is mostly localized to the periphery of the nucleus. And we care about this because not only is nuclear architecture cool, but we know that increasing evidence shows that it's important to development, especially at the peripheral environment. And we can see that in several different papers, such as this one here, talking about the importance of the peripheral environment in C. elegans development, as well as pre-implantation biology and human embryonic stem cells. Additionally, increasing evidence is showing that in addition to being involved in development, that the peripheral environment is also involved heavily in human diseases. A classic example of that are the laminopathies. So those tend to be muscular dystrophies. We also, though, see defects in the nuclear periphery and several different types of cancer, as well as aging diseases such as progeria being one of the more common ones. The Epstein lab has studied the nuclear periphery for several years now, and one of the big things that we have focused on is a histone modification known as H3K9 dimethyl. So that is the dimethylation of the 9th lysine residue on the H3 histone. And one of the reasons that we care about that is because, as you can see from this storm image here, H3K9 dimethyl marks the nuclear periphery. And that may be even more apparent if we zoom in close to the edge, where you can see the edge of the periphery being marked in red here by lamin B. And adjacent to that, you can see all of this green H3K9 dimethylated chromatin that is adjacent to that nuclear periphery. And this is really a unique aspect of H3K9 dimethyl as compared to some of the other transcriptional repressors that you might see at the nuclear periphery. So for example, we find H3K9 trimethyl as well as H3K27 trimethyl also at the nuclear periphery. However, unlike these other marks, which are more ubiquitously distributed throughout the nucleus, H3K9 dimethyl is really centrally localized to that nuclear periphery. So when I joined the lab, I was really interested both in learning more about H3K9 dimethyl, but also the enzymes that are responsible for catalyzing its deposition. And the enzymes responsible for catalyzing most of the H3K9 dimethyl that we find in a nucleus are known as GLP and G9A. And they work together as homodimers to be able to deposit dimethyl on our histones. And that, as I mentioned previously, results in transcriptional repression. However, if you lose GLP or G9A, they're no longer able to catalyze the deposition of H3K9 dimethyl, and that results in aberrant transcription. And we know that in humans, that defects in the gene that encodes GLP can lead to a disease called Kleefstra syndrome. Now, this is a congenital disorder. It's very rare, but it does result in several congenital defects, such as craniofacial defects, as well as congenital heart diseases. So when I first started thinking about GLP and Kleefstra syndrome, one of the things that kind of first became apparent to me was that the tissues that are affected in Kleefstra syndrome are frequently tissues that we also see that are derived from the neural crest. And we had a great introduction into the neural crest earlier today, but there are several different types of different populations of neural crest that contribute to different parts of a developing embryo. So for example, we have our cranial neural crest, which is going to contribute to the frontal bones of the face, as well as our cardiac neural crest, which is going to contribute to the great vessel development, as well as making a contribution to the semilunar valves. And then of course, there are other neural crest populations which are not going to contribute to tissues that are known to be affected in Kleefstra syndrome. And so as I was thinking about that, it really led me to my first question, which was, could I use the neural crest as a model in mice to have mice that develop Kleefstra syndrome-like phenotypes? So to be able to investigate this, I took advantage of mice that were flocks for the genes that encode GLP and G9A, and then I crossbred those with mice that carried CRE under the Wnt1 promoter. So that allowed me to target the ablation of both of these proteins specifically in the neural crest population. Very recently, I have actually also been able to breed in a lineage reporter. So I have TD tomato in the Rosa 26 locus. And some of you may be familiar with lineage tracing. So what that means is if there is CRE present in these cells, so if my two GLP and G9A are ablated, then those cells are also going to express the tomato. So they and their progeny are going to be red, whereas cells that are non-neural crest cells will remain normal. So after I started breeding these mice, one of the first things that I really wanted to do was to ensure that I was getting rid of these proteins in my mice, because nuclear proteins are often difficult to wash out of these animals. It takes a couple of cycles after you've cut out the genes to actually dilute the proteins out of the animals through proliferation. So in all of my slides, I'm going to try and have some sort of illustration or schematic. I know a lot of people aren't used to looking at mouse embryos, so you will see either here in the middle or over on the side an embryo with a plane to show you where we're looking. And so in this case, we're looking at an embryonic 11.5-day embryo here. And we're looking through the craniofacial mesenchyme, which is a large proportion of neural crest derived cells. And as you can see by looking in our green channel here, GLP, when we look at our control versus our double knockout, we can see that GLP is not being expressed by our neural crest population. And that's perhaps even more apparent if we take a little bit more of a zoomed in view. And the same thing I found was true for G9A. So the double knockouts in the neural crest are not expressing GLP or G9A protein, which was great. My model was efficiently able to get rid of these proteins in early embryonic development. I also wanted to check into the ability of these mice to still have H3K9 dimethyl in my neural crest population. So what you're looking at here are some immunohistochemical stains, again, of an embryonic 11.5-day embryo. And we are zoomed in to a place called the otic vesicle. That's where your inner ear is going to form. And this is a great place to look before you have a lineage trace actually bred into your mice, because this consists of a ring of non-neural crest cells that are sort of embedded in this larger sea of neural crest-derived mesenchyme. And so when we take a look at our control, which is over here on the left, and our double knockout on the right, what you'll notice is that H3K9 dimethyl, which is here in pink, is broadly expressed in our non-neural crest population and our control, but is largely absent from our neural crest population, which just went to show that indeed I was able to functionally change the H3K9 dimethyl in these embryos with a knockout of GLP and G9A. And surprisingly, this actually didn't affect other transcriptional repressors such as H3K9 trimethyl, as you can see here. And this was sort of a surprise because in the literature, H3K9 dimethyl and trimethyl are often sort of lumped together as one big unit. It'll even be written out H3K9 ME2/3. And that's because a lot of people thought that you had to go through dimethyl to actually get to trimethyl. And so this piece of data here is actually just a little bit of evidence that these are truly unique modifications, H3K9 dimethyl and trimethyl, that are being regulated by separate enzymes. And that was a really cool finding for us. So once we had established the model, I was really interested to know if my mutants were going to be born in normal Mendelian ratios. And indeed they were. So at postnatal day 0, we observed and collected several different litters of pups, and we found that we had double knockouts in the expected ratio. However, we did observe that at the time of collection that several of the pups were already deceased. And this was especially true if 3 of the 4 G9A and GLP encoding alleles were depleted in these animals. When I examined the surviving pups, what I noted was that these animals were likely deceased because of a failure to feed. And you can observe that by looking at the white milk spot here on the lower left abdomen of our control and see that that's absent here in our mutant animals. Upon further histological examination, I found that these animals were likely not feeding because of the presence of cleft palate. So just to walk you through the slide and what you're looking at, these are Mason trichrome images of a control over here on the left and a double knockout on the right. I've highlighted the palatal shelf here for you with a red box. And I think what everyone can appreciate is that the control has a nicely fused palatal shelf, whereas the double knockout has failed to fuse. And this was a highly penetrant phenotype in these mice. And once again, when 3 of those 4 G9A and GLP alleles were depleted, it was almost 100% penetrant. In addition to cleft palate, these mice also had dysmorphic skulls. So one of the first things I noticed when I was doing my dissections was that my double knockout animals, which you can see here along the bottom, were microcephalic. In addition to having small skulls, the double knockouts also had a underdevelopment of all of the bones that were derived from the neural crest, which I've highlighted for you here with these blue arrows. Now, it is important to note that the non-neural crest-derived bones, such as the ribs or the tibia, did not seem to be affected. They had normal numbers of ribs, and the tibia lengths were not statistically significantly different. So in addition to having these craniofacial defects, my mutants also had defects within their cardiovascular system, the most prevalent of which was patent ductus arteriosus. So the ductus arteriosus is a vessel that runs when you're a fetus between the pulmonary arteries and the descending aorta. And the purpose of this vessel is to direct blood to the fetal circulation for oxygenation. However, when we take our first breath of life, smooth muscle, which is derived from the neural crest, closes off that vessel, closes off the ductus arteriosus, and shunts blood to the lungs for oxygenation. So in my control animal here, you can see that the ductus arteriosus is nicely closed. However, when we look at our mutant, you can see, and this was a mutant that was collected alive and was breathing prior to dissection, and you can see that this mutant has an open ductus arteriosus. And again, this was a highly prevalent phenotype when 3 of the 4 G9A and GLP alleles were depleted. And I want to just take a little bit of time to take a closer look at this, because I had the pleasure of working with some amazing summer interns this year, and I wanted to highlight a little bit of their work. So they were able to take a lot of photos and actually align histological sections and then draw out these 3D reconstructions using a program called Imaris. And you can see the control 3D on the left here and the double knockout on the right. And then we're just going to take a quick look through these histological sections. So what you will see is the presence of the ductus arteriosus. And you can see throughout the length of that vessel that it is completely closed off. However, in our double mutant, what you'll see is that that ductus arteriosus does remain open, fully connecting the pulmonary arteries and descending aorta. So in addition to ductus arteriosus, these animals also had several rare valve defects. I'm a valve person through my PhD training, so I can't ever resist just taking a moment to talk briefly about the valves. And so what you're looking at here is an H&E image of a normal aortic valve. The aortic valve is a tricuspid structure. So it has 3 cusps that make up the body of the valve, and that's the left coronary cusp, the non-coronary cusp, and the right coronary cusp. However, one of my mutants had the presence of a supernumerary cusp, so a 4th cusp. This is a very rare phenotype. I've never seen this before. It was really, really exciting when I saw this. And then a little bit more common, I had a couple of mutants that had the presence of a left non-coronary fusion. So they only had the presence of 2 cusps or a bicuspid aortic valve. And again, I'm just going to highlight the work of my summer interns here, and we'll take a look through these histological sections. You could see all 3 of the valves or the cusps present in our aortic valve and our quadcuspid valve. We have the left coronary, the non-coronary right coronary, and then there's the supernumerary valve. And just one more, the bicuspid valve. You see the left and right coronary valve, but you never see the presence of a non-coronary cusp come into play. So this was really cool with all of these different phenotypes, but it really left me with the question of what mechanism of normal neural crest development is actually being affected by GLP and G9A. And I don't have time to go through all of the data that I've been able to collect so far, but I want to give you guys just a quick taste of what I think is going on. So those of you that are familiar with the neural crest know that there are really 4 big possibilities that could be going on in these mutants. We could have a deficiency in the proliferation of these cells. They could be apoptotic. They might not actually be migrating to the correct places in the face, or they may be having a issue with their differentiation. And I have looked at proliferation through Ki67 staining at several different stages. These are E11 brachial arches, but I've also looked at E13, E15, and postnatal day 0. And none of these time points are there any differences in that are statistically significant in the number of proliferating cells. So we can kind of go ahead and knock that one off the list. The same thing can be said for apoptosis. So I've done a bunch of different cleaved caspase 3 staining at various different stages, and there do not seem to be any differences in those sections as well. So for now, we're going ahead and knocking out apoptosis as well. I've shown you several pictures of these craniofacial mesenchyme, and you've seen that the red cells are definitely getting where they're supposed to be going. There's a ton of red cells. I've also done a little bit of quantification. This is still an ongoing work because I need to get true wild types to actually test, but of the genotypes that I've had available with tomato to be able to count the number of cells, there doesn't seem to be a difference in the number of cells that are present in either the craniofacial or the cardiac region. So I also don't think it's a defect in migration. And so that really leaves us with differentiation. And this is definitely very new. I actually just sent off some samples for RNA sequencing. So I'm hoping to get some really good answers for differentiation, but just to give you kind of a taste of what I think is going on. You can see in these embryonic day 11.5 embryos that a protein known as MEIS2 is not being as readily expressed in our double knockouts. And I've highlighted some box sections here to just try and show you in these brachial arches from which the palatal shelf will protrude in just a couple of days. We can see that there's a downregulation of MEIS2, which is upstream of the ossification pathway for forming bone in the face. And if we look just a couple of days later at embryonic day 13.5 at this MEIS2 expression, you can see here in the palatal shelf, which I've highlighted here below, that the MEIS2 expression is dramatically reduced in our mutant animals compared to the control. And again, these are pathways that are upstream of RUNX2 and ossification. So just kind of giving us a little bit of a hint that the cells that are getting, the neural cells that are getting to the palate are not differentiating properly into bone. And so just to give you kind of a really broad view of a model for what I think is actually going on with GLP and G9A, in their presence, they are able to add H3K9 dimethyl to different genes and enhancers. I mentioned to you that that is a transcriptional repressor that can relocalize different loci to the nuclear periphery for transcriptional repression. However, if you don't have GLP or G9A, they're no longer able to catalyze the deposition of H3K9 dimethyl on these aberrant transcriptional loci, which means that we're getting transcription that shouldn't be happening. We're really confusing our neural crest cells. And so there's an inhibition of their normal differentiation pathways. And with that, I would just like to thank you all so much for your time and your attention. I'd like to thank the members of my lab that helped to contribute to this project, my collaborators, of course, my 2 fabulous summer interns that I had, and then I wouldn't be here at all without my funding sources. So I would like to say thank you, especially to Additional Ventures. And if any of you are on Blue Sky, I'd love to connect with you, and I'm happy to answer any questions.
Thank you so much, Diana, for the very amazing talk. So we have a question from Crystal Rogers. So this was so cool. So many new crystallographies are represented here, highlighting the clear importance of neural crest cells. So do the double mutants, they also have abnormal PNS, ENS formation or patterning, or just phenotype in the cardiac neural crest and the cranial neural crest?
Yeah, that's a really great question. Right now, the only defects I have seen are in the cardiac and craniofacial structure. So I haven't seen any defects in any of the other structures. I've actually looked at some of the truncal and sarcal neural crest as well for like the distribution of ganglia. I've looked at the adrena medulla, and there don't really seem to be any differences in the differentiation or expression of normal things in those populations.
I see, thank you. So this is just my follow-up question. So do you have any guess or hypothesis that why the knockout of G9A is only affecting like a subpopulation of neural crest cells?
Yeah, I've been thinking about this a lot lately, actually. It's something that's kind of been tickling my mind. And I read a paper recently that talked about all of the different signaling that goes on, especially in the face. There are such small regions that have very distinct patterning in craniofacial development. And so my hypothesis is that because you have all of these different signaling pathways that are also going to contribute to different organ formation as well. So that you might have like BMP signaling that is not only important for craniofacial development, but also other organ development, that it's more important to lock down, if you will, to transcriptionally silence these alternative fate loci in the craniofacial environment where they have all these different signaling, as opposed to, for example, the truncal environment where there isn't quite so much signaling going on. There's more of a consistent pattern. So that's my hypothesis at this point, but it still remains to be seen.
Yeah, thank you so much. And we have another question from Angelo Iliana. So the question is, are there any of the double knockout mice viable long enough to measure blood flow from the heart? It's curious to know how it flows in the very odd 4-valve mutants.
So they are not living very long at all. So only a couple of hours after birth. I maybe could get something within that time period, but they're definitely not making it to adulthood. However, I am really interested. So, I breed with heterozygote males. So I am curious, even if those males, they do live and they're able to reproduce, if they might be sensitive to certain perturbations. So perhaps like they don't have, the big heart defects that I showed that are going to lead to lethality, but perhaps they might be more prone to like cardiomyopathies if they're challenged. So that is something that I'm very interested in looking into in the future.
Thank you so much. And we have another question from Talia Hakovich. Nice talk. Do you see any signs of transdifferentiation in the double conditional knockout? Are the cells taking on a different identity?
So that is why I actually sent out for single cell RNA-seq from E11.5 craniofacial mesenchyme. So I'm really hoping to get those data back quickly, because I do think that what I'm going to see is expression. I don't know if it's going to be, so my question is it going to be stochastic, where we just have these random genes that are coming on, or are there going to be certain pathways that are more likely to take place? So for example, instead of following a bone differentiation, are they going to differentiate into like adipose or to cartilage or something like that? So that's something I really think will be more apparent when I get the RNA-seq data back.
Thank you. And we have a question from Pierre Van Vliet. Love your sessioning in IF. The mutant valves look thicker and hyperplastic, yet other neural crest defects seem to be a lack of tissue. And because proliferation was not different, what do you think will be the cause of the hyperplasia and the 4th valve leaflet?
So that's a really interesting question as well. And I know, so in the craniofacial mesenchyme, I know that there is an importance for hyaluronic acid, and that is also something that is very important to valve development. You have to have that cushion for the cells to be able to invade. And so I'm wondering if there may be some sort of connection with the production of hyaluronic acid. So defects in the way that the ECM is being laid down rather than the number of cells in particular. So I think that that may be at the root since it's in both locations, but again, still need to be determined.
Thank you. And we have another question from Matthew Staffield. Tubercle system. Can you speculate a bit more on how the loss of a repressor may lead to the downregulation of a key neurocrest transcription factor, for example, the MIS2 that you mentioned? What could be the molecular intermediate here?
Okay, and so I didn't quite catch all of that question. They were basically asking what is happening in the nucleus, I think.
Yeah, because I think the rationale could be like if you knock out the G9A, then you would expect like a lot of genes get upregulated. But like what you showed here is like there's a loss of the MIS2, like the expression is lost. So what could be the reason behind this?
Yeah, so I suspect that the ability to express abnormal genes, so genes from alternative lineages, is actually causing the cells to downregulate the normal genes that they would express. So I have a feeling that these cells are actually transdifferentiating into another cell type, or are just remaining more neural crest, more mesenchymal. So that's my suspicion, is that the overproduction of other genes are actually causing the repression of the normal expression that we would see in neural crest. Great question, thanks.
Yeah, and we have another question from Hashi Kumati. Awesome work. Since the H3K9 dimethylation is important for repressing transposable elements, I'm curious if you have ever seen or are you planning to test whether there's a repression of transposable elements in these mutant nuclear cells?
Yeah, that's a really great question. So the heterochromatin is definitely full of all sorts of really cool transposable elements, signs and lines, and we have looked. I didn't go into, so there's a whole bioinformatics part of this project that I didn't go into that'll hopefully be actually getting published soon. But there are different types of H3K9 dimethylated chromatin, and we find that they're modifying different TEs depending on which type of chromatin it actually is. So there's a type of H3K9 dimethylated chromatin that actually interacts with the nuclear lamins, and then there's another kind that doesn't seem to have that same interaction. And that type of chromatin that is lacking that interaction with the lamin B seems to be full of like transcriptional enhancers and like sign elements, for example, whereas the other type of chromatin that has that lamina interaction is got more of your, for example, your lines.
Yeah, very interesting. So we have another question from Kerry Adler. The localization of the dimethylated H3K9 at the nuclear periphery is really striking. Have you looked at nuclear morphology in your knockout by EM or other markers besides the trimethyl stingling to see whether there are any other global defects?
So I haven't done that yet, but that's definitely on my to-do list as something I'm very interested in looking at. I want to see if there are consistent morphology differences in the nucleus, because especially in the heart, H3 or heterochromatin in general is known to be at the nuclear periphery to help provide structure, because there's all those hemodynamic and shear forces that are experienced by cardiomyocytes, for example. And so I think that this dimethyl layer actually is going to end up being pretty important to providing stability to a nucleus. So that is looking at EM is definitely something I would want to do.
Thank you so much. So we have many questions, and and thank you for the great talk for today. And also thanks Joshua for sharing with us your science. The seminar has been recorded and will be available on the SDB website next week. Please join us for next month's seminar on Friday, April 11th, when Talia Hackfridge from the Duke University and Gerald Loderick from UC Santa Cruz will present. Thank you all for coming. See you next time.